Welcome everybody to Question of the Week. This is a Facebook Live session that I'm going to be pushing to my Facebook page every Monday morning at 9:30 a.m. on the Pacific Standard Time and 12:30 in the afternoon for those of you on the East Coast. And in this Facebook Live session, I'm going to be taking on your science faith questions. For those of you who frequent my Facebook page, you know that every time I post an article or a podcast or a video link, there's quite a bit of discussion that takes place. People offering comments, people asking questions, and of course I also get a lot of questions through private messages, as well as questions that people post on my page. And I try to respond to as many of those as I can, but try as I might, I just simply can't do it. I have just a limited amount of time. So the hope here is with this question of the week is to be able to take on one of those questions that I cannot get to, that I think is an intriguing question that would be of general interest, and tackle that through these Facebook Live sessions. And if you are on my Facebook page, you know that we have quite a bit of discussion that takes place. There are people who offer opinions that complement my post, other people who offer dissenting opinions, and that's perfectly fine. And so if you disagree with something that you hear me saying during my response to this week's question, please feel free to comment in the comment section below, to air out your perspective, to disagree with me as you would like. I think it's a lot of fun to have those kind of conversations that take place around very important questions, in my view, about the relationship between science and the Christian faith. If you want to check in during this broadcast, please feel free to do so. I see that several people are already checking in. It's a lot of fun for everyone to see who's listening live and where you're from. So please go ahead and feel free to do that. If you want to post a question in the comment section for me to consider for next week, also go ahead and do that. And of course, throughout the week, please feel free to private message me with your question or to post the question directly to my Facebook page. So anyway, without much further ado, I'd like to go ahead and get started taking on this week's question. And this week's question is from Eric Elvgren, who last week in the comments section of the Facebook Live session posted his question that deals with miniaturization and the case for complexity in biological systems. And Eric's question is this: in modern technology, small size seems to be synonymous with more advanced technology. Smaller chips and so forth seem to be the goal. Does biology consider size to be significant in determining complexity? So a really, really interesting question that actually leads us to a very powerful case for God's role in bringing life into existence at its most basic fundamental level, the biochemical level. But before we tackle the question formally, I just want to acknowledge that when I posted this topic for this week's question of the week, my friend Andy Grell, who quite often will post on my Facebook page, offered this insightful comment about Raquel Welch and her miniaturization that took place during the Fantastic Voyage movie. And so, Andy, this question has nothing to do with Raquel Welch and the Fantastic Voyage. But anyway, all kidding aside, let's go ahead and take a look at this week's question. Biologists probably are not very quick to recognize any relationship between miniaturization and complexity, but there is a group of scientists who do, and those are scientists working in nanoscience and nanotechnology, typically chemists, material scientists, and engineers. And the reason why is because this area represents the ultimate in miniaturization, where scientists are trying to build machinery, devices at a molecular scale, where they're trying to assemble molecules into these complexes that have structures and functional capability that allow these ensembles, these molecular ensembles, to perform specific tasks. And this process is an extremely difficult process that is challenging, to say the least. But it's also a very exciting area of technology where there's very real potential to lead to a new revolution in terms of our technological capabilities if we are capable of miniaturizing things to a molecular scale. But in order to pull this off, these scientists are forced to actually study biochemical systems. Why is that? Because biochemical systems are essentially nanotechnology. These are machines inside the cell that are formed through the ensemble, through the aggregation of molecules, and these molecular ensembles, these chemical supersystems in the cell, carry out specific tasks. In fact, they work in conjunction with one another to perform supra-tasks that then contribute to the overall biological activities that are necessary for life to exist at a cellular level. And because, in a sense, biochemistry is nanotechnology, scientists working in nanotechnology and in nanoscience are essentially studying biochemical systems to help them understand the principles that they need to drive miniaturization to a molecular scale. And I think this is highlighting that relationship between complexity and biological complexity and miniaturization. In fact, many times these scientists are not only turning to these systems for inspiration and insight, they are literally mimicking the designs of biochemical systems to implement new nanotechnologies, and in some cases they are actually co-opting those designs, isolating the molecular systems from the cell and interfacing them into the nanodevices that they are actually trying to build. Just a few examples to illustrate some of the way in which biochemistry is intersecting with nanotechnology. There are these protein complexes in the cell that are referred to as biomolecular motors. They're massive protein complexes that literally have an architecture that is identical to the machinery that we would build as human designers. One example of hundreds that I could point to is the F1F0 ATPase. This is a rotary motor that functions as an energy transduction machine that takes electrical gradients across membranes, which stores energy, electrical energy, and will convert that electrical energy through the flow of electrical current that passes through what are called motor proteins into a rotation. And that's achieved through the motor proteins interfacing with a rotor that's made out of proteins. And as that rotor rotates relative to a stator made out of proteins, the rotation is translated through a drive shaft made out of proteins with a cam at a right angle that crashes into protein turbines in a series of protein turbines in a circular manner, and the mechanical displacement of those turbines drives chemical reactions that stores that electrical energy in the form of chemical energy that can then be used by the cell's operation to power different processes in the cell. But this is an example of, again, the type of nanotechnology we see in the cell that then gives insight to nanoscientists as they try to design basically rotary motors from scratch, building them from individual atomic components. But there are some nanoscientists who have gone one step further, and instead of using the molecular motors as inspiration to power motion or movement in these nanodevices, they are actually isolating them, purifying these machines, and interfacing them directly into nanodevices, again co-opting the designs in nature to solve a problem in miniaturization. Another area where we see this intersection between biochemistry and nanotechnology that again highlights this theme of miniaturization and biological complexity is with regard to the DNA molecule. DNA is a molecule that stores information in its structural makeup, and that information that is stored is digital information. There's also ways in which DNA actually harbors analog information, but that's a whole nother conversation. But that digital information that's stored is information that the cell's machinery uses to make the protein machines that we just talked about. These instructions are not only telling the cell's machinery how to make those proteins, but when to make them in the cell cycle, and how many copies of those protein machines to make. And of course, one of the big challenges confronting us today is a data storage crisis. We're producing more data than we have the capacity to store. So people are looking at how can we miniaturize storage capability, and DNA is a beautiful example of the ultimate miniaturization with maximum storage capacity. In fact, people estimate that a gram of DNA could store as much information as found in 10 trillion compact disks. Some people have estimated that 1 kilogram of DNA could store all the data that we have generated throughout human history. So the storage capacity of DNA is incredible, and there are people that are now exploring how we can actually utilize DNA to store information, and some proof of principle experiments have been done that have shown, for example, we can encode the entire contents of a book into DNA and actually read out that content in a controlled manner using techniques in molecular biology. So here's an instance where we're co-opting again a miniaturized technology in the cell to solve our engineering problems. But there are also scientists who are using the inspiration and the insight regarding the way in which DNA encodes information to try to design novel polymers that can store digital information that are much more reader-friendly or user-friendly for our applications, not for the applications in the cell, but for our applications. And so here's an instance where biochemistry is inspiring nanotechnology. And to me, the most fascinating example of all is something called DNA computing, where scientists are literally building computers using DNA as digital information and the enzymes that manipulate DNA as what are called Turing machines. And for those of you who know something about computer science, a Turing machine is an abstract machine that was invented by Alan Turing, the father of computer science, that forms a theoretical basis for how computer systems function. And it turns out that the machinery that manipulates DNA in the cell is literally functioning like a computer system. And so scientists are now trying to take advantage of that fact and build DNA computers using, again, DNA and the enzymes in the cell that manipulate DNA. And these computer systems are housed in little tiny Eppendorf test tubes that are about this big, and they're more powerful than the most powerful supercomputer system because of the possibility of employing massive parallel operations simultaneously because of the nature of the molecular system that they're dealing with. But here's, again, another example of a design in biochemistry fueling technological advances in nanoscience and nanotechnology. So in summary, what we're saying here is that, yes, indeed, there is a relationship, in my view, between miniaturization and biological complexity, and that relationship is not evident to most biologists, but becomes clearer when you think about biochemistry in light of nanoscience and nanotechnology, where the inspiration and the insight for building nanodevices comes from studying the way in which biomolecules interact with each other, because these are nanotechnology. These are nanosystems that are much more sophisticated than anything that we could hope to build. Or scientists are mimicking or copying the designs in biochemistry to build new technology, or in many cases, they're just simply co-opting the cell's chemistry and utilizing it in a technology context. But all of this work not only, I think, highlights this relationship between miniaturization and complexity in a biological context, it segues to, I think, a very powerful case that we can make for the role that a creator must play in bringing life into existence. Biochemical systems are the most basic fundamental systems of life, and we see a very clear signature for a creator's handiwork in the nature of biochemical systems. The fact that these systems are so similar to the types of technologies we build in terms of motors and machines and digital storage devices and computer systems, it reinvigorates the watchmaker argument that William Paley advanced in the late 1700s. William Paley in the late 1700s argued that a watch, which he described as a contrivance, bears the fingerprint of a creator, a watchmaker. And when he compared a watch to biological systems, and he was studying macroscopic systems, he noted a similarity between those systems and a watch, and he argued from analogy, if a watch requires a watchmaker, then biology must require a divine watchmaker. A very powerful yet simple, straightforward argument that I think has given new life, given the similarities that we see between biochemical systems and, again, human technologies. Now, this watchmaker argument has been challenged on two fronts. One is the Humean challenge, which basically relates to the use of analogies to argue for, in this case, a creator's role in bringing life into existence. Hume argued, and rightly so, that when you're engaged in analogical reasoning, the conclusion that you derive is only as sound as the similarity of the systems you're comparing. The more similar the systems, the more sound the conclusion. The more dissimilar, the less sound the conclusion. And Hume and others who followed after him have argued that a watch and a living organism are so different that what we conclude for a watch doesn't necessarily apply to a living system. And in fact, there are philosophers of biology like Massimo Pigliucci who have argued that when we talk about machines in the cell, we talk about information in the cell, this is just simply a metaphor that scientists are using. We shouldn't conflate that metaphor with true information and with true machinery. But when we begin to look at biology in light of nanoscience, it's very clear that these protein machines are machines because we're using them in some instances in nanodevices as machines. That the information in DNA is digital information because we're using it that way in our technology. That DNA and the machines that manipulate DNA in the cell are really computer systems because we're using it in that way. And so the application of biochemistry to nanotechnology takes care of, I think, this Humean critique of the watchmaker argument. And then the other critique, of course, is that is represented by Darwin and the evolutionary paradigm, where the claim is that evolutionary mechanisms can produce basically the designs that we see in biology. And there's a number of ways that we can challenge that claim, but one of them, again, relates to this idea of nanoscience and nanotechnology, because scientists are turning to the designs in biochemistry to inspire developments in nanoscience and nanotechnology. And this, number one, highlights the elegant designs that we see, but I think is fundamentally incompatible with the nature of the evolutionary mechanism. Because according to evolutionary biologists, evolution's mechanism is an unguided, undirected mechanism that has no end goal or no foresight whatsoever. It is a historically contingent process that co-ops existing designs and cobbles them together to produce new designs. And that kind of mechanism is going to produce evolutionary kludge jobs, suboptimal designs that are just cobbled together. They may be fine-tuned to some degree and optimized to some degree because of the influence of natural selection, but fundamentally, at their very essence, these designs are kludge jobs. So why, in their right mind, would any scientist or engineer turn to designs in biology repeatedly if they're cobbled together, if they're suboptimal designs, to inspire new technology? That makes no sense. Yet we are comfortable doing that because these are elegant designs, and they're so elegant that the elegant nature of these designs seems to be incompatible with the mechanism evolutionary biologists claim generate them. And it's nanoscience and nanotechnology and this quest for miniaturization that I think really shines new light and new perspective on the sophistication and the ingenuity of biochemical designs. And so what we have here is a multi-layered, multi-tiered argument for design thanks to nanotechnology. It invigorates the watchmaker argument, and it helps us to respond to the Humean and the Darwinian challenges to the watchmaker argument in such a way that I think we have an incredibly compelling case for God's role in bringing life into existence. And this is one of the reasons why I'm so committed to the idea of intelligent design, why I'm so committed to the idea of a creator's role in orchestrating not only the origin, but the history and the design of life, because of these insights that are coming from emerging areas in science and engineering like nanoscience, nanotechnology, and also advances that are happening in synthetic biology. But that's another topic for another day. So anyway, that brings us to an end of this session. Again, question of the week, where I take on your science faith questions. If you want to ask a question again for me to consider for next week, go ahead and post that question in the comment section of this video, or you can private message me or post the question on my page. Anyway, it's a pleasure to see so many of you listening to this broadcast live. I'm also excited about those of you who will listen to this broadcast as a taped video feature that's available through my Facebook page. Thank you for your interest in our ministry here at Reasons to Believe and the things that we do. We very much appreciate you. And I'm just going to leave you with this final thought, that the more that we know about science, the more that we have reasons to believe. So thank you so much. I'll see you next week.